I haven't sprayed off anything yet, so that's of a concern to you. You gotta give me a minute. Sarah. Yeah. Got your code. Thanks for logging in. 2, 3, 4, 5, 6. Speaker go off. Hi, everybody. I get the speaker up, we can say hi to one another. Hopefully you're willing. All right, folks at home, I would love for you just to turn your camera on briefly and give me a hi or say hi to your classmates, just so I know you're okay and you're alive. And if you want,
Classmate.
Hi, good morning.
all right. And folks here in the classroom, hi, real quick.
Hi.
Hello. Hello. Not bad, right? Just like a normal classroom, normal school year. Let's just keep pretending that it's normal, right? It ain't normal. Not even a little bit. Let's call it what it is. All right. Well, welcome, and I'm glad you're here, no matter where you are. I have a check-in. I'm hoping it's going to be a little bit better than the last class. There was an issue that we may have the same issue. We're going to find out here momentarily. But I thought we could spend maybe half the class looking at the biochemistry of fats, because we didn't have a lot of time during our time together last week to review that. So I've got some practice questions that we can do that might prompt some questions from you. Of course, you may be totally fine with all of it, and that's okay too. And then the second half of class, we can begin to build a deeper understanding of what I hope you had a chance to watch in the Edpuzzle video on the first of a two-part video on protein structure, which is where we're going next in terms of the biological molecules. So carbohydrates, and then lipids, and then proteins. It's really the sequence that we're trying to follow here. And again, I've got some practice questions that will give you a chance to build a deeper understanding. All right. But before we get started, as always, questions, comments, anything for the good of the group. Okay. Well, I say it a lot, but if something comes up, please don't hesitate to ask. Let's see if my check-in will work better. No, still not working like it's supposed to. There's supposed to be different emojis, not just the one. So don't drag your, unless it applies to you, you don't have to drag your dot to the one sad face. But it's kind of how I'm feeling today. So if I could drag my dot, that's where I would be. I'll fix that for the rest of the classes this afternoon. I'm sorry about that.
It's the only option.
It's not supposed to be the only option, I promise. There are others, and there's a continuum. So I'll have to fix that for later. I'm not sure what's going on. Probably a paired up thing. But I'm hoping that you're okay. Even if you're not okay, I'm hoping that you know what to do to take care of yourself and the people that you care about. Because this is what we used to call crunch time, right? End of the semester, final exams, 2 weeks-ish to go. Take care of yourselves, especially if you're not feeling so great right now. I'll just say really, really briefly, if it gets to a point where you feel like you can't do what I or other teachers are asking of you, say something. Okay? Don't let your grade, your scores, your status in college admissions dictate how well you take care of yourself right now. No, there's nothing big next week. I mean, to be honest, Maya, they can't see what you wrote, but it's to everyone, so I hope it's okay to share. I have written one more take-home assessment. Okay, so number 6. It's not live yet, it's not due yet, and we're just going to kind of take it day by day. If it feels like it's appropriate to do that before the end of the semester, we will. If not, we can save it for later. All right, but no, nothing big. I hope there's nothing big in this class, period, right? That it all feels manageable, small-ish. And if not, you're going to tell me, because I'm willing to modify it if it is. Seriously? When I heard the authors project was still a thing, my jaw dropped a little bit. Could it be like authors project light? But I get it. Old habits die hard, right? All right. Well, let's at least follow our agenda minus the check-in, which I'll fix with other classes today. I said we'd start with fatty acid, with lipid, with fat structure, and then do that for the first half, and then for the second half of class, we can begin to work with protein structure and function. Actually, before I even do that, let me go back just a little bit to our agenda. Again, I hope you look at these agenda slides, whether it's the day of class, where they pop up, or when you go through the notes to look at them on your own. It's a pretty big one that we've been focusing on now for the last couple of class periods. And it's this idea, that color's not gonna work. It's this idea that the subcomponents of biological molecules and their sequence, right, the order in which they appear, determine the properties of the molecule. Okay, so it's so important that I'm highlighting it now, but much of what we're going to do today is built around that very idea. So each of these molecules are fatty acids. So they would be part of a larger fat if they were all put together. And I've got a short video clip that will show us how those fats might be put together. But what's important to us in terms of the subcomponents of these molecules is that, I pull my pen up, a molecule like this, is this a saturated fatty acid or an unsaturated fatty acid? Saturated. There's no carbon-carbon double bonds that are part of this structure. And if you're wondering, how Jesse knows that, every point of intersection here, it's kind of like shorthand in chemistry, is a carbon atom. Now the hydrogen atoms aren't shown, again, it's shorthand, it's like a skeleton structure, but they're there, and I could draw a few of them in. But there's no carbon-carbon double bonds that are part of this structure. So this is a saturated molecule. But if I go down below, there's only one other type of fatty acid that's relevant to us. This molecule, with its carbon-carbon double bond, how might we classify it? It's an unsaturated fatty acid. We could go a little bit further. This is going to be the topic of this video clip. It's a mono unsaturated fat. There's only one carbon-carbon double bond. But based on its configuration, it's a cis mono unsaturated fat. And if you look at the structure of these two molecules as they're depicted on the screen, the saturated fatty acid, it's pretty linear, right? It's pretty straight. Where in the unsaturated fatty acid, there's a bend. It kind of looks like a V or a kink shape. It's going to be super important to us as we consider how the subcomponents, right, the presence or absence of carbon-carbon double bonds, and even how those carbon-carbon double bonds exist as structural isomers of one another, impact the properties of those molecules as a whole. Okay? That's what this little video clip is all about as I get ready to play. Before I play it, questions or wonderings on your part about these molecules? Monitoring the chat too, folks, so if you've got things you're wondering about. Yes, pleasure.
So are unsaturated fats the ones that are liquid at room temperature?
Yes.
They can't pack together?
Right, which is where we're going next. Yes. And let's be really clear that while it's fine, right, to quote unquote memorize that unsaturated fatty acids are liquid because they don't pack together, you can have unsaturated fatty acids that do pack well together and are not liquids at room temperature. And that, of course, as I get this ready, is the topic of this video clip. So I'm going to try to make sure you can hear everything. And folks at home, if you can't hear it all right, let me know. But hopefully everybody, no matter where you are, will be able to see and hear this short summary of the different types of fats and why they matter.
Olive oil is 100% fat. There's nothing else in it. Pancake mix, on the other hand, is only about 11% fat. And yet, olive oil is good for you, and pancake mix is not. Why is that? As it turns out, the amount of fat we eat doesn't impact our weight or our cholesterol or our risk of heart disease nearly as much as what kind of fat we eat. But let's back up. What is fat? If we were to zoom in on a salmon, which is a fatty fish, past the organs, past the tissues, into the cells, we would see that the stuff we call fat is actually made up of molecules called triglycerides, and they are not all alike. Here's one example. Those 3 carbons on the left, that's glycerol. Now you can think of that as the backbone that holds the rest of the molecule together. The 3 long chains on the right are called fatty acids, and it's subtle differences in the structures of these chains that determine whether a fat is, let's say, solid or liquid, whether or not it goes rancid quickly, and, most importantly, how good or how bad it is for you. Let's take a look at some of these differences. One is length. Fatty acids can be short or long. Another, more important difference is the type of bond between the carbon atoms. Some fatty acids have only single bonds. Others have both single and double bonds. Fatty acids with only single bonds are called saturated, and those with one or more double bonds are called unsaturated. Now, most unsaturated fats are good for you, while saturated fats are bad for you in excess. For saturated fats, the story pretty much ends there, but not for unsaturated fats. The double bonds in these molecules have kind of a weird property. They're rigid. So that means that there are 2 ways to arrange every double bond. The first is like this, where both hydrogens are on the same side and both carbons are on the same side. The second way is like this. Now the hydrogens and carbons are on opposite sides of the double bond. Now, even though both of these molecules are made up of exactly the same building blocks, they are 2 completely different substances and they behave completely differently inside of us. The configuration on the left is called cis, which you've probably never heard of. The one on the right is called trans, and you probably have heard of trans fats before. They don't go rancid, they're more stable during deep frying, and they can change the texture of foods in ways that other fats just can't. They're also terrible for your health, by far worse than saturated fat, even though technically they're a type of unsaturated fat. Now, I know that seems crazy, but your body doesn't care what a molecule looks like on paper. All that matters is the 3D shape, where the molecule fits, where it doesn't, and what pathways it interferes with. So how do you know if a food has trans fat in it? Well, the only sure way to know is if you see the words partially hydrogenated in the ingredients list. Don't let nutrition labels or advertising fool you. The FDA allows manufacturers to claim that their products contain, quote, 0 grams of trans fat, even if they actually have up to half a gram per serving. But there are no hard and fast rules about how small a serving can be, and that means you'll have to rely on seeing those key words, partially hydrogenated, because that's how trans fats are made, by partially hydrogenating unsaturated fats. So let's go back to our olive oil and pancake mix from before. Olive oil is 100% fat. Pancake mix is only 11% fat. But olive oil is mostly unsaturated fat, and it has no trans fat at all. On the other hand, more than half the fat in pancake mix is either saturated or trans fat. And so even though olive oil has 10 times as much fat as pancake mix, it's healthy for you, whereas pancake mix is not. Now, I'm not pick on pancake mix. There are lots of foods with this type of fat profile. The point is this. It's not how much fat you eat, it's what kind of fat. And what makes a particular fat healthy or unhealthy is its shape.
All right. So some review in there, but also I thought some really nice illustrative examples of why this matters. Is it bringing up any questions for you? Pass the chips? Pass the processed foods? When I say pass the bag, or pass, I don't want to eat it, right? It's the big question. And it's tough. It's tough because those foods are delicious. They're manufactured to be delicious. You heard the narrator say very briefly with a little drop of saliva coming out of the character's mouth, the cartoon's mouth. The trans fats provide even a savory mouth feel, like this feeling of unctuousness, like a melt in your mouth feel, which is desirable. So, you know, we've been led by our food manufacturers to crave the very foods that are not good for us. So, oops. So the subcomponents of biological molecules influence their properties, right? And there can be subtle differences in the structures of these molecules that make it so that in the case of cis fatty acids with their bent structure, those molecules for the most part are not going to be able to stack very well. And really, they're not even going to be able to interact very well because of their three-dimensional structures, where in the case of trans fats, still unsaturated, their linear structures are going to allow them to stack, to interact in ways almost analogous to that of a saturated fat. Okay, so let's try, not this, a little bit of practice. First, let's just see if we can identify some different fatty acids by dragging your dots. You gonna be able to see those? Okay. Let me see if I can fix that real quick. Ah, that might be why. Let's see. Give me a second here. I should be able to fix this. Huh. So you can see the agenda, but you can't see anything else. All right. Okay, let's try this. Everything is slowing down. Almost there. Come on, buddy. Almost there. Still not showing up? What if you just refresh your page? So all you're seeing is the agenda, right? Is that true for everybody?
Yeah.
Okay, we're gonna make it work. It's okay. If you want to take a short break, stand up, stretch. It's like everything is not wanting to work. You can always just present it too. It won't be quite the same. But I can do that if I have to. Connected. Oh, wonderful. Still not going. Okay, now it's moving, right? All right, let's get to the right slide. Just moving slow as molasses, that's all. I'll get you there. Goodness gracious. It's always wonderful when things slow down to a crawl. Okay, looks like now you can see it. All right, sorry about that. So again, the task is a simple one. You've got these different fatty acid structures. Just trying to identify which one is which. by dragging your dot. Oh, no. No, stay. Are you okay? I think I've got 9 of 12 folks. Okay, let's take a look. See how you did. How we're getting there. Okay, kind of a mixed bag. So if you're going to drag your dot, are you going to drag your dot because everybody else did or because you know where to drag your dot? All right, looks like maybe taking a moment to talk about these would be helpful. So I'm going to hide your responses here. And our first fatty acid, of course, we have this carboxyl group sitting out here, C double bond O OH. Okay, it's one of the key distinguishing features of a fatty acid. We have one carbon-carbon double bond, but it looks like it is in the trans configuration, right? That if there's a carbon here, and here, and here, and here, these are skeleton structures again, that those carbon atoms are on opposite sides of the carbon-carbon double bond. So this would be our yellow up here, a yellow dot. Okay, we'll just kind of move around clockwise. Let me know if you have questions. Another carboxyl group sitting over here. No carbon-carbon double bonds. This would be saturated. This would be our red. Write this in. That's saturated. This is going to be cis monounsaturated. Okay. And then here we've got another carbon-carbon double bond, but this time carbon, 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 carbon. These two carbon atoms, or if you prefer, of course, there are hydrogen atoms that aren't shown in the diagram. They're on the same side of that carbon-carbon double bond. So this is a cis monounsaturated molecule. So just a little bit of practice differentiating between cis and trans fatty acids. And then last but not least, we know this has to be our polyunsaturated. There's more than one carbon-carbon double bond. But let's see here. Let's start with that one. Cis or trans? Trans. Nice. How about this one? Cis. This one. Remember, there'd be a carbon here and a hydrogen and a hydrogen. There'd be a hydrogen here too. What do you think? This two would be cis. Yeah. And this one's a little bit trickier, right? Because there's a carbon sitting up here, but there isn't a carbon sitting down here on the other side. Okay. So this would be a cis as well. All right. So good. A little bit of practice identifying the different fatty acids. But why does it matter is the next logical step. So this problem probably looks familiar to you, but take a moment. 2 different fatty acids, linoleic, palmitic acid. One clearly is polyunsaturated, one is saturated. Which one do you think is a solid at room temperature? About halfway there. There we go. This is an AP multiple choice question. All right, let's take a look at what you came up with. Yes. Yeah, so palmitic acid, it is the saturated fatty acid molecule. Remember that the linear molecules, because of their structure, they're going to be able to pack closely together. They're even going to be able to form weak intermolecular attractions between one another that just aren't possible between unsaturated fatty acids like linoleic acid. And as a consequence, those molecules are going to be solids. They're going to have higher melting temperatures, and the other molecules are going to be liquids. So one last set of practice questions, just thinking about this. Now I've got 4 different fatty acids. They're all depicted in the same way, so there's no clear giveaway in terms of which one is which structurally. But can you identify them based on their structure? Or you could, hypothetically, identify them based on their melting temperature. But I'll say that all of the information associated with a particular color dot, it all goes together. So if you think something is polyunsaturated, it is, in this example, a cis polyunsaturated fatty acid with a melting temperature of -11 degrees Celsius. About halfway there again. Try to get your response in, even if you're not 100% sure. There's no penalty here. Nobody knows what you're choosing. The only person who could know is me, and that's only if I drill down to that level. I have better things to do with my time, I promise. All right, let's see how you did this time. Things still moving around a bit. Okay. Looks like there's a consensus, maybe 1 and 3, a little bit uncertain. Would it be helpful to talk about it just for a minute? Folks at home, thumbs up or some indication that it would be helpful? Okay, thank you. All right, so again, this one's a little bit trickier, right? Because they're all drawn in the same way. There's no bends or kinks or linear molecule structure that would give it away. So let's kind of move clockwise again. Let's see, we've got carbon-carbon double, carbon-carbon double. So I've got 2 carbon-carbon double bonds there. None here, well, that's a giveaway. That makes this molecule saturated. And because it's saturated, it's going to end up with the highest melting temperature. It doesn't even seem that hot, but we'd have to heat it to that temperature in order for the substance to be converted from a solid to a liquid. Then we've got 1 carbon-carbon double bond there. Okay, that makes it easy. That's a monounsaturated molecule. So it's going to have a relatively low melting temperature, in this case 13 degrees Celsius. So it's going to be a liquid at room temperature. Remember, room temperature, do you have a sense of what that is in centigrade? It's okay to say no. What's room temperature in centigrade? Do a lot of work with that number in chemistry? Yeah, it's usually 25 degrees Celsius. So this would be a liquid at room temperature. And then 1, 2, 3 carbon-carbon double bonds. Well, that's a giveaway. This is my polyunsaturated fat. So it's going to have the lowest of low melting temperatures, right? It's kind of funky. Negative 11 degrees Celsius is the melting temperature. So yeah, duh, it's going to be a liquid. In fact, I'd have to cool it to negative 11 centigrade to convert it from a liquid to a solid. Okay. But what does that mean for the others? Well, this is going to be a polyunsaturated, but it only has 2 carbon-carbon double bonds. So in terms of its level of bendiness, right, of kinks, it's going to be slightly less bent than number 3 would be. So we would expect it to have a slightly higher melting temperature, still pretty low, right? Still -5, but higher than -11. And that's going to make that final choice monounsaturated. Okay. So all this is ultimately going to matter in terms of the molecules that are present in our bodies, that are present as part of a living system, because they're going to influence the membranes that serve as the boundaries of all of our cells. So what I'm hoping you'll begin to see as we come back to this later on in the year is that if you had a cell membrane that was composed of a lot of saturated fatty acid, or even trans unsaturated fatty acid, you're going to have a structure that's more rigid, that's more solid-like than you would if you had a membrane that was composed predominantly of unsaturated, whether they're mono or polyunsaturated fatty acids, which will allow your membranes to be much more fluid and flexible. It's going to be super important to us in so many different ways in respect to how molecules get into and out of cells. It's all going to be based on the structure of those membranes. But for right now, structure contributes to the physical properties, the physical property being melting temperature. Questions before we move forward? We've got just a little bit of time left to practice with what you saw in the video that, again, I'm hoping you watch before coming to class today. Because we're moving on to the third type of biological molecule. We've done a little bit of work with carbohydrates, some more work today with lipids and fatty acids, the building blocks of lipids. And if you had a chance to watch the first part of this 2-part video on protein structure, you were introduced to proteins as the third biological molecule. So with that in mind, again, let's do a little bit of practice. This is a multiple choice question from a release AP exam. I'm hoping you're noticing the generalized structure of an amino acid, the building block of a protein, the monomer that comes together with other monomers to build the polymer, which is the protein. Which part of this particular amino acid do you think is non-polar, is hydrophobic? Looks like everybody has a response. All right, we have a leading vote getter for sure, but not everybody is in agreement. Would it be helpful to talk about why that methyl CH3 group is the non-polar / hydrophobic group? I'm seeing some heads nod here in the classroom. Folks at home, would that be helpful? Thank you, Max. Thank you, Maya. The smartboard never raises its thumb. Disengaged smartboard. All right. So again, what I'm hoping you're noticing when you look at a picture, again, the video is the lesson, right? You need to watch these videos before you come to class so that we can use that information and practice with it. You can always go back and watch the video again, but the video is the lesson. It is the content. It's the reading if we had a textbook, okay? So it's super, super important that you're doing that before you come to class. All right, so we have a generalized amino acid here. Actually, we have a specific amino acid when it comes right down to it. But all amino acids have a central carbon atom. And to that central carbon atom, we're always going to find a hydrogen atom. switch colors, an amino group, switch colors one more time, a carboxyl group, and then finally, one more color switch, a side group. Sometimes an author will say R group rather than side group. So in this amino acid, the side group is CH3. Okay. Well, you might remember from the Edpuzzle video on the functional groups that just about every functional group that's relevant to biological molecules is a polar functional group. How do we know? Well, the nitrogen atom here is more electronegative than either of the hydrogen atoms. Holy chem review. You with me? Doing okay? People, when they hear the word chemistry, they have nightmares or tremors. Okay, but nitrogen's in group 5. It's further to the right on the periodic table, certainly further to the right than hydrogen. So again, I'm going to do something. I hope it looks familiar, but if it doesn't, let me know. The nitrogen atom would have a slight negative charge, that del symbol, delta symbol, just means a partial charge. Where over here, the hydrogen atoms would have a slight positive charge because they're less electronegative than the nitrogen is. So you could even get in here 2 dimensions, but we have a net dipole pointing towards the nitrogen atom. So that part of the molecule is polar, short story long. If I come over here to the amino group, excuse me, to the carboxyl group, once again, oxygen more electronegative than carbon. Hydrogen less electronegative than carbon, so there is a bond not depicted here, but it's holding the O and the H together. So in a case like this, I have a net dipole pointing, again, 2-dimensionally in this direction, but this functional group is also polar. Okay? It's a difference in charge spread out across the molecule. These bonds, oops, that didn't work. They're all polar covalent. Electrons are not being shared equally between the atoms. If I were to redraw the CH3 part of the molecule down below, just expanding it out a little bit, again, justifying our answer, okay? Look, all these carbon-hydrogen bonds, the difference in electronegativity between carbon and hydrogen is negligible, okay? These are not polar covalent bonds that hold the carbon and hydrogen atoms together. They are non-polar covalent bonds. For all intents and purposes, the electrons are shared equally between those atoms. So collectively, this functional group, this methyl group, is non-polar or hydrophobic. Those 2 terms are interchangeable. Okay? Does that help? Head nod, head shake, something indicating that the words coming out of my mouth are being heard. Thank you. Okay. Well, all of this matters to us as biologists, right? Because when you look at an amino acid, all the parts of an amino acid, except for that side group, they're the same. In fact, when you put amino acids together, they are the monomer of a protein. To build a larger polymer, the only part of the amino acid that isn't interacting in the formation of those bonds is the side group. And since there are 20 different amino acids, there are 20 different side groups, each with their own physical properties, which you need to memorize in order to be successful in this class and in life in general. He once again says sarcastically. Because if I need you to use this, right, I'm going to give it to you, as will the College Board. Okay? But take a look real quick before we sign off. Here's, now you probably can't see it. Sorry. Here's, oh my goodness, stop it. That CH3 group. Notice it's part of a group of amino acids that are all non-polar. Why? Because while the rest of the amino acid is the same, the side groups are different, and all of these side groups, well, They're all composed, in this case, of carbon and hydrogen atoms. They're all nonpolar. They're all hydrophobic. But there are other amino acids that have different physical properties, right, as a consequence of their structures, and are going to behave differently, especially when we start assembling all of these amino acids into larger proteins. Okay? So, sorry. Before you take off, if you would, just quickly, as we think about the work that we did today, scroll to the bottom. Okay. Whether you're looking at a fatty acid or an amino acid, the subcomponents of these molecules determine their properties. How are you feeling about this? You starting to see the connection? You definitely seeing the connection? Or is the connection really unclear to you right now? Just drag your dot. Okay. All right. So with that, once you've dragged your dot, that is our exit slip for the day. We'll pick up on this on Wednesday. So folks at home, you're welcome to sign off. Folks here in the classroom, I'm going to come spray the surfaces. For Wednesday. Okay.